Hi, I'm SUDENUR from Turkey. Today I want to present my project and you can see the title on the screen. First of all, I want to talk about cancer. Actually, as you know, cancer is a very deadly disease and cancer comes second after cardiovascular illnesses and human mortalities. In this project, my objective is to find out which Curcumalonga and Propolis dosage are more effective on vascular formation in egg embryos. That is, this study discusses the preventive impacts of Curcumalonga and Propolis on vascularity in egg embryos. The effects on cancer will be studied by observing which dosages of these substances to be used are effective on vascularity to what extent. Now, I want to give a little information about Curcumalonga and Propolis. Curcumalonga plant is now to have been used in Indian medicine for a long time in the treatment of flu, cough, liver disorder, rheumatism, sinusitis, and anorexia. Curcumalonga used as food colorist is colorless, heat-resistant, and composed of tetrahydrocurcumin, which is antioxidant. Propolis became now in the treatment of disease 1,000 years ago, and it was used by Egyptians, Greeks, and Romans as a natural remedy during the cure of many illnesses or palliative purposes. As I mentioned before, I worked on egg embryos, and now I'm going to talk about the development process of chick embryos. This is very complicated and spans around 21 days. In this process, there are 2 stages and external interventions such as medication and injection. Application shouldn't coincide 42 hours and 20 days of their growth. The cardiovascular system starts from at 25 hours. Angiogenesis is the process of the formation of blood vessels, and it can be controlled with chemical substances, for some of which repair the damaged blood vessels and stimulate the formation of new ones. Now you can see the dosages of Curcumalonga and Propolis to be injected into chicken egg were prepared. 4 different dosages were prepared for each substances. From Curcumalonga, dosages of 20, 40, 60, and 100 milligram milliliter are used, and from Propolis, the dosages of 0.1, 0.2, 0.3, and 0.5 milliliter are used. Now I'm going to tell you how I make a mixture. To extract the curcumin active substance from Curcumalonga and dissolve Curcumalonga, 10% ethanol was prepared and 10 milliliter ethanol and 214 milliliter distilled water were mixed to get 250 milliliter mixture. Then this mixture was sterilized in autoclave, and this mixture was diluted with distilled water. 15 gram Curcumalonga was added into alcohol distilled water solution obtained to get extract substances released into the mixture. It was left to cool at room temperature. After the cooling process, it became ready to use. The eggs to be used had embryos and were newly fertilized, and they were used on the same day. The extract accurate was injected into eggs, and the opening front was covered with wax. After being kept in incubator for 96 hours, vascularity in the egg was tested. The experiments were repeated 3 times and analyzed whether the results were correct or not. At the end of 96 hours, the eggs taken from incubator were broken and embryo development and vascularity were examined. It has been found that in correlation with the increase of curcumin dosage, the vascularity formation in embryo decreased. In control group, in the eggs into which 1 milliliter distilled water was injected, there were heartbeats at the end of 96-hour growth. As to experiment group, the vascularity decreased in parallel with curcumin dosage increase. As to Propolis experiment, it found that at the beginning the vascularity decreased in parallel with curcumin dosages increase, but later it was observed that Propolis had a toxic effect on the embryo. In the Propolis dosages of 0.1 and 0.2, the vascularity decreased, but in the dosages of 0.3 and 0.5, the embryos died as a result of toxic effects of Propolis. Because of this reason, Propolis should be used carefully. Findings have proved that both Curcumalonga and Propolis not only have significant effects on vascularity, but stop the formation of blood vessels as well. During my literature review, I didn't come across any publication about the effects of Propolis on vascularity. In my study, I haven't only investigated the Propolis on vascularity at first time, but I have also reached conclusions that it can inhibit the angiogenesis formation in cancerous tissues at the end of experimental tests. Thank you for your time, and if you have any questions, I will try to do my best to answer them. Thank you.